Good evening, everyone, and thank you for joining us tonight. My name is Melanie Wooden, and I'm Dean of the Faculty of Arts and Science. Before I begin, I wish to acknowledge the land on which the University of Toronto operates. For thousands of years, it has been the traditional land of the Huron-Wendat, the Seneca, and most recently, the Mississaugas of the Credit River. Today, this meeting place is still home to many Indigenous people from across Turtle Island, and we are grateful to have the opportunity to work on this land. I'd like to welcome you all to the 2022 Martin Lecture in Ecology and Evolutionary Biology. And welcome to our guest lecturer this evening, Professor Andrew Reed, Director of the Huck Institutes of the Life Sciences at Pennsylvania State University. In a time of continued uncertainty, I look forward to hearing his insights on the trajectory of COVID-19. The Faculty of Arts and Science created this lecture series to recognize the Martin family's exceptional generosity. We feel extremely fortunate that the Martin family has maintained deep ties with the University of Toronto through the years and generations. All 4 of Mary and Ron Martin's children graduated from the university, as did all 6 of their grandchildren. In fact, when we went as far back as the late 1800s, we counted some 60 members of the extended family who all attended U of T. The Mary and Ronald Martin Graduate Scholarships, for which we are deeply grateful, are a fitting way to honor this family's legacy. These awards truly speak to the Martins' passion for learning and their commitment to supporting the next generation of scholars. As many of you may know, the Martin Graduate Scholarships help us attract brilliant students from across Canada and all over the world. They are awarded to top graduate students in 4 disciplines: Astronomy and Astrophysics, Ecology and Evolutionary Biology, Physics, and Public Policy. And the annual Martin Lecture Series rotates in focus among these 4 departments. This year, we are excited to host our 3rd Martin Lecture, and the first one in Ecology and Evolutionary Biology. This department is at the forefront of research into the factors governing biodiversity, from genomes to ecosystems. Home to 3 Canada Research Chairs and 3 Fellows of the Royal Society of Canada. This department's thought leadership has helped the university establish a reputation for excellence in biological sciences research. And now, I will turn this over to Professor Stephen Wright, Chair of the department, who will introduce our keynote speaker. Stephen?
Thank you, Dean Wooden. So it is my great pleasure tonight to introduce you to Professor Andrew Reed. Originally from New Zealand, Professor Reed moved to the UK to pursue a PhD in evolutionary biology at the University of Oxford. After holding a number of prestigious fellowships, he became the chair of natural history at the University of Edinburgh, a professorship that was established in 1767. In 2007, he moved to the Center for Infectious Disease Dynamics at Pennsylvania State University, where he is Pugh University Professor in Biology and Entomology, Eberly Professor in Biotechnology, and Director of Penn State's Huck Institutes of the Life Sciences. Over the past 3 decades, Professor Reed has established himself as a leading voice in the study of the evolution of infectious diseases. He has authored more than 250 peer-reviewed publications, 30 book chapters, and 4 edited volumes. His research investigates the evolutionary forces that shape pathogens, as well as those that shape the immune responses of hosts. He's made major contributions to understanding the evolution that's prompted by our medical and public health measures, such as drug and insecticide resistance and vaccine escape. Not content with understanding the negative outcomes of these control measures, Professor Reed has dedicated himself to the challenging goal of designing health measures that are less prone to erosion by evolution. In a nutshell, can we design strategies that are evolution-proof? To do this, he combines mathematical modeling, lab-based experiments, and increasingly analysis of clinical data through close collaboration with medical doctors. For his contributions to infectious disease evolution, Professor Reed has been elected to fellowships from the Royal Society of Edinburgh, the American Association for the Advancement of Science, the American Academy of Microbiology, the American Academy of Arts and Sciences, and the Institute of Advanced Studies in Berlin. And just before I invite Professor Reed to speak, just a note that there is an option for viewing the transcript of the session using the Zoom button at the bottom of your screen. And also we encourage people to ask questions. You can submit questions during and after the lecture using the Q&A button, and we will have a question period after the lecture. So with that, we are delighted to host Professor Andrew Reed as our speaker for the 2022 Martin Lecture in Ecology and Evolutionary Biology. And I invite Professor Reed to give us his lecture. Thank you.
Thanks very much, Professor Wright. And I really appreciate the great honor this is to speak here tonight. I want to talk about the, whoops. Hopefully that's good for everybody. I want to talk about the future of COVID-19, but specifically where the virus itself is going to go next. So what will the next variant or variants look like? And I want to acknowledge that I have had a terrific amount of support over the years from various agencies to fund the work that I'm going to draw on tonight, which became very, very timely since the pandemic started. So this is the phylogeny of where we've got so far in terms of the virus evolution. So the virus pops up or spills over in China at the end of 2019. And then since then, there's been rampant evolution of the virus. So we've had the alpha variants, we've had beta variants, gamma, delta, and delta, of course, has gone crazy recently. And then most recently, Omicron, which of course has caused the most recent problems. And note that Omicron was churning away for a long time before it popped up and became a problem. But that's the past. And what I want to do tonight is to talk about the future, and in particular, whether or not what the future variants might look like. And so the questions I want to deal with are, will there be new variants? And of course, there will be new variants. Nothing stops evolving. So there will be new variants. What we really care about is variants of concern, ones that would affect human health and well-being. And if there will be variants of concern, what will they like? Will they be like? And I think that really boils down to two questions. Will they erode the vaccine or infection-induced immunity in people? Will they be able to get around that immunity that people have? And will they be able to cause more or less severe disease? And these are questions about predicting the future of evolution. And predicting the future, especially if you're an evolutionary biologist, is a very nerve-wracking thing. So I want to say right from the outset that the crystal ball is extremely hazy here. And that's important because there's no simple way to make predictions in evolutionary biology. In fact, evolutionary biology as a science is essentially advanced hindsight. We're very good at explaining, I think, what happened after the event, but we're not very good at going forward in time. And evolutionary biology is essentially a historic science, and it's a lot like human history. You know, people can give very good accounts of how something came to be in history, but it tends to be after it's come to be. It's very hard to predict in advance. Now, my physicist colleagues give me a very hard time about this. They say, well, we just write a few equations and we can go to the moon. And I just want to spend a few minutes saying why evolutionary biology is not like that. It is not rocket science. And I think, although it might be a lot cheaper than going to the moon, in a sense, going to the moon is a pretty easy task, figuring out how from Newton's equations we can hit the moon. But that is not what evolutionary biology is about. And that's for a couple of reasons. One is that there is a lot of chance and contingency that goes on. So chance happens because mutational processes are a roll of the dice. Combinations of mutations, which ones appear in the same virus, for example, is a roll of the dice multiple times. And then we can get recombination of lineages where, for example, the Delta variant might recombine with the Omicron variant, so they swap bits of their genome. But that requires that both Omicron and Delta would be in the same person at the same time, and that they would end up in the same cell, and that the right processes of recombination would happen. And so there's a lot of chance and sort of ticking of the clock, lottery tickets involved. So that's one of the reasons why it's hard to predict. There are also events that matter a lot. And so, for example, a variant that gets on an airplane from South Africa and gets to Canada, the first variant out of the country will be a very significant impact on what happens next. And in fact, the spillover from whatever yet to be discovered into humans, of course, was an event which didn't need to happen. A lot of things had to line up for that particular genotype to spill over and start to spread in the human population. Super spreader events are another example where big parties can create massive events that spread variants through a population. So there's a lot of chance and contingency that goes, that means we have a lot of trouble predicting what's going to happen next. But it's not just that. The other side of it is, for something that's novel, we don't know what variants are possible. The only way we know something is possible is once it's arisen. For example, I don't think anybody could have conceived of something like Omicron a few months, maybe a year or two ago, at least. It's hard to know what's possible before something's actually appeared. And just to give you an example of that, if you think about a parrot, you probably think about something like this. If you think about parrots in general, how many people could imagine a parrot that's 4 or 5 times as big as this one, that walks on the ground, that doesn't fly, that only eats low-grade nutrition and only breeds once every 4 years, and even then, the only time the males and females come together is for sex? That is not your average picture of a variant of a parrot, and yet parrots like that exist. And this is a very young picture of me holding such a parrot. These are kakapo from New Zealand. Prior to the discovery of these birds, nobody could have conceived of these birds, no matter how many birds you'd seen before. And likewise, before people went to New Zealand, there was nobody who imagined a bird like this. Once you've seen them, it's easy to imagine how they arose. We know what the kiwis do and why they're there and so forth. But predicting in advance that a kiwi could happen is an extremely problematic, very, very challenging task. And here's another one that's fascinated me most of my career. We know wheels are the most efficient way to move around a landscape. You know, bicycles are the most efficient means of turning energy into movement. And so we ought to expect animals to evolve wheels. But we clearly don't find any like this. And why is that? Well, we think it must be very, very hard. And that's because animals have been around for a long time. So you'd think they could have stumbled upon it by now. And it could be that there aren't materials that animals could produce that could rotate. So maybe it's an engineering impossibility. Maybe there still hasn't been enough time for the right combination of mutations to come together. Or maybe half a wheel, an imperfectly formed wheel, is worse than a leg. So maybe you just can't get from legs to wheels because in between stages are just dysfunctional. So you can speculate about that simply because we know that these animals, such animals, don't exist, but they should. And so you can sit around thinking about a virus and imagining what it might do, and it becomes very problematic when you've got a completely novel virus. And I think one of the things that's so special about SARS-CoV-2 is that we have never experienced a pathogen that spilled over into humankind before, and so is rapidly adapting to us, getting better and better at infecting us, while simultaneously being vaccinated against. So we're in completely new territory here. And that is kind of like, well, we've set out and we're going to go find out what's on New Zealand. What sort of birds are there? We're going to look around the world for the predicted animal with wheels. It's very, very new terrain and very, very hard to make predictions. And so this is the spike protein of SARS-CoV-2. And these are the mutations, the red blobs are the mutations we now know can happen and are structurally sound and appear in variants. But in March of 2020, we had no idea which mutations could appear. And this is but one of the 30 or so proteins that the virus produces. And we don't yet know what's going on in those other 29 or so proteins. We don't know what mutations are possible there and how they might combine with these ones to give different phenotypes. And so when things haven't appeared, we're stuck really with imagining what can go on. And I've been engaged in that process with viruses before. In 2000 or early 2000s, when SARS-1 came and then burned out, we tracked it down and eradicated it. I remember being at a conference where we sat around, with beers after the conference discussing what sort of virus might actually persist and be more harmful than SARS-1. And then we started thinking, what would be the scariest type of virus you could get? We all agreed it would be respiratory, it would be aerosol transmitted. It wouldn't be as lethal as SARS-1 because that's pretty easy to track down when people are getting really sick and dying. It would have a very large amount of pre-symptomatic transmission, so transmission before anybody knew they were sick, and maybe a large number of asymptomatic cases. And I can remember sitting around the meeting saying, could a virus do that? Is it possible for a virus to do a lot of transmission before it makes someone sick? And of course, we know there are mild viruses, but mild viruses don't worry us. Could there be viruses that can do a lot of transmission before somebody gets sick and dies? And we had a long discussion about whether such a virus could exist. And I remember vividly thinking it would be really scary if viruses like that were possible. And 20 years later, we know they're possible. So it's very difficult to make predictions in advance. And I want to emphasize that substantial humility is needed by us evolutionary biologists. And that's not always been the case. And I actually see on Twitter most days people telling us what's going to happen next. I just, for context, want to put up this paper. It was published in March of 2020, so just after the virus was really spreading, although the preprint was posted in February. And we shouldn't worry about viruses mutating. If you look at that second sentence, in reality, mutations are a natural part of the virus life cycle and really impact outbreaks dramatically. And I put it to you that if we ever thought that was a wrong statement, it's following Delta and Omicron. Mutations have substantially, dramatically affected this outbreak and our happiness and well-being. These authors are some of the biggest names in viral evolution. I don't actually personally know Nathan and Mary, though I know them professionally. They're at Yale. Eddie Holmes I've crossed paths with him my entire career. He is one of the biggest names in virus evolution. And I gave him a hard time about this paper a few months back, and all he said was, yes, it didn't age well. So with that and the substantial humility noted, I'm going to have a go at saying where I think things are going to go in the future. Even though it's hard to predict, there are a couple of things that we can work off to try to make predictions. And these come from the fundamental understanding that we get from Darwinian natural selection. And I want to make the point that Darwin's birthday is in just a few days. From Darwin's work and everything that's followed through, we know that we can work off two axioms. The first one is that natural selection always favors variants that, in the case of viral infections and transmission, are variants that have a transmission advantage. Okay, and in this case, with a pandemic, we mean the number of infections per infection per unit time. So variants that have an advantage where they are able to transmit more than one new infection per infection will spread through a population. And that is the pressure we know that's always there. If there's a variant that can pop up that has a transmission advantage, it will spread. Okay? A variant that can spread, well, it will spread. And one way of looking at that is R0, which is a measure of the number of new cases per case. The estimates are that when we first got the variant in 2019, the original one, it had an R0 about 2 or 3, which meant that it could infect 2 or 3 people for every case. Delta, the estimates are that it infects 6 to 7 people for every new case. We know that Omicron is greater than Delta. So they've had, each one of these variants has had a transmission advantage over the previous ones, and so they've spread. And just to put that in perspective, that we haven't yet seen anything as transmissible as measles, which has an R0 of around about 15. I do want to come back to those numbers. The actual numbers themselves, I think, are increasingly irrelevant. They are estimates of the number of new cases per case in a fully susceptible population, and we no longer have fully susceptible populations. Anything, though, that has positive transmission advantages, so will infect more than one case per case, has the potential to spread. The other thing we know is that we can't have the Darwinian demon. And the Darwinian demon is an organism that has infinite fitness. So in this case, it would infect infinite number of people as soon as a person got infected. We just know that transmission to an infinite number of people is not possible, and it's not possible to do that immediately. There's got to be time for the virus to work itself up, and then there's never going to be enough transmission before the infection is cleared or the person dies, as much as there could be. So there has to be some limit, some upper bound on how good these variants can be at spreading. And those are really two axioms. There's a bound, and under that bound, any variant that has a transmission advantage will spread. Okay, so with that background, I want to say, will there be new variants of concern? I would say there's absolutely no reason to think that Omicron is the final word. I think there's every likelihood, and in fact, within the Omicron lineage, we already see a second lineage that's starting to spread. It's possible that Omicron is the final word, that's the pinnacle of fitness for the virus. It's very, very hard to imagine that's the case now. And I remember answering journalist questions, is Delta it? Is Delta it? Saying, I don't think Delta is the final word. This thing's got more. And then we got Omicron. And I think one reason to think that it can't be the final word is simply that the world is changing very fast as far as the virus is concerned. So this is data out of South Africa. So the purple line is the number of new cases per day in South Africa, and the green line is the number of vaccine doses administered. And the first part of that plot, the first wave is where Delta dominated, and the second wave is where Omicron dominated. And you can see that Omicron faced a very different world from Delta. Delta was facing people who were largely unvaccinated, and it was spreading in a largely susceptible population. Omicron, on the other hand, is spreading in a world that was largely vaccinated, or at least substantially more vaccinated, and had just had immunity generated by Delta from the people that got Delta infections. So the world that Omicron saw and spread in was a Delta immunized, vaccine immunized world, whereas Delta spread in a world which had maybe seen Alpha and Beta before. And so very different picture is emerging all through time. So the virus itself is not seeing the same world that its predecessors saw. And so a variant that might not have done very well early on could do better now in a world which is different from where it didn't do very well early on. And so this changing world means that we have not got anywhere near the final word because the world is changing continuously. Let me give you another example that I've worked on extensively. This is a virus of rabbits that was released in Australia. When Australia was settled by Europeans, they had the crazy notion that it would be good to release rabbits in Australia as a fur industry. And so that worked fine to start with, and you can see the number of fur pelts there. But very soon the rabbits did what rabbits do, and it was covered Australia and turned a lot of it into a dust bowl. So the rabbit fur industry became the destruction of agriculture. And as a result of that, the Australians looked around for a virus that might be able to take out the rabbits, and they found myxomatosis in South America. And in 1950, depending on your view, it either accidentally escaped or was deliberately released to control the rabbit populations in Australia. And this virus spilled over into this rabbit. So the South African rabbit that it was found in is a very different rabbit from the European rabbit that was in Australia. And incredibly, within 6 months, the virus had spread over 1 million square miles, and tens, perhaps hundreds of millions of rabbits died in that first 6 months. This is the biggest amount of death known for any disease outbreak in a vertebrate. Absolutely incredible. It got over that space, 1 million square miles in 6 months, and perhaps 100 million rabbits died. So that's a spillover event to beat all spillover events. We've been working on it extensively, and the we here is the Eddie Holmes I mentioned before, Peter Kerr, Isabella Catadore, and that's some of our experimental work at Penn State, which I'm going to mention in a minute. But this is a phylogeny of that virus since it was released or spilled over. This is the ancestor that got out in 1950, and then this is the virus as it spread through time. Now, the branch lengths here of the phylogeny of the tree of rabbit life are representative not of time, but of the amount of genetic distance. And so in here, we have isolates from the 1990s, from 2016, 1990s, and 2016. So the virus has been spreading and evolving just like we see with SARS-CoV-2. and continues to do so today. These viruses that we got from the field in 2016. The amazing thing is that this lineage C, has actually got longer branch lengths, so it's more genetically distinct. The virus has sped up its rate of evolution. So 70 years or so after the spillover, the evolution is still going crazy, and in fact, it's escalating. Now, we don't know why it's escalating, but the only real way you can explain an escalation is that the viruses in lineage C have some sort of trick that allows them to spread more rapidly, evolve faster, and that's what we're seeing. So that's a really good case example where even after 70 years, there's no way we've exhausted all the evolutionary potential of that virus. So I'd be hugely surprised if Omicron is the last variant we see. The virus is continuing to spread and the world is changing fast. And it's not just the types of vaccines that are being used are different in different parts of the world. There are people who've never been infected. There are people who've been infected once. There are people who've been infected with 2 variants, 3 variants. The world is continually changing for this virus, and it's very hard to imagine that Omicron is the best variant at this rapidly changing world. And I don't think there's any reason to think that evolutionary potential, the amount of mutations, the combinations, the recombinations can run out at this point. So variants of concern will continue in my view. Okay, so what will they be like? Well, I think the first question there is whether or not they're going to undermine vaccine immunity, vaccine-induced immunity. And this is a timeline since the 1920s through to more or less the modern day, comparing the fate of drugs, so antimalarials and antibiotics, with these are human drugs, with human vaccines. And you can see that antibiotics, so this is penicillin, for example, went into widespread use around the middle of the Second World War, and resistance was first seen in British hospitals in 1945. Tetracycline discovered shortly after the war, resistance first seen in British hospitals in the mid-1950s, and so on. So resistance rapidly evolves to drugs, and you can see that's always been true. In some of them, it's frighteningly short before you get the resistance appearing. Whereas these are the human vaccines, and actually the vaccines, some of them have been, for example, diphtheria, have been never undermined by evolution. So no variants have come along that are able to get through a diphtheria vaccinated person. And indeed, smallpox proved so unable to evolve around the vaccine that it went extinct in 1979. So it was unable to find an evolutionary solution to the smallpox vaccination. And in fact, if you look down the lists, the purple ones are viral, the green ones are bacterial. It's actually the case that by and large, the vaccines have held extremely well. They have not been undermined by the evolution of the target pathogen. Even the ones that have are rare, and pertussis is, even though there's some variants that do better in vaccinated people in pertussis, the vaccine itself is still very successful. Likewise for pneumococcus, those were changed slightly to deal with that, but not a big catastrophe. In hepatitis B, there are variants that can get out of a vaccinated person and into other people, but they seem not to spread very well in a population for reasons that aren't clear. So by and large, the picture for human vaccines has been that evolution of the pathogens, new variants have not undermined vaccination. And a colleague of mine, Dave Kennedy, and I reviewed those cases where variants have appeared, and the most remarkable thing to me was that even though there are variants that are able to get around the immunity, the health benefits associated with vaccination have largely been sustained. So even though the pertussis vaccines, there are variants that are able to do better in vaccinated people, nonetheless, the vaccines still stop whooping cough very, very effectively. And the disease symptoms associated with those different pathogens are still substantially suppressed by the vaccination. And so in general, whenever we've had a situation where variants have spread so that they are able to get around the vaccine-induced immunity in the sense of getting into people and getting out of them, the vaccines still prevent disease, so people getting sick and so forth. And data coming in for especially good data from Ontario, actually, but also elsewhere in the world, seems to show that for Omicron too, that the Omicron is very able to get into people who've been vaccinated and get out of them. It's very able to get into people who've got immunity, so they've seen the virus before, get into those people and get out. Breakthrough infections seem very, very common, but vaccinated people are much less sick than unvaccinated people. So far, the variants that we're seeing are kind of like the ones we've seen elsewhere in human diseases, that the anti-disease vaccination, anti-disease immunity is very strong and holds up despite these variants, even though they might have failed in the sense of, or failing or reduce the chances of infection, but not to zero. So the way I think about that is if you have a vaccine efficacy measure, and then we've got fitness on this axis, so this is infections per infection per unit time. When we got the original strain pops into the world, when we get our first vaccines, they're very effective against the initial strain. And then we got Alpha and Delta, where the protection dropped slightly in terms of infection. So the vaccines were slightly less likely to stop a person getting infected, but they still worked very well against severe disease. And then this is a plot I actually drew up before Omicron, wondering where it was going to go next, what would happen. And of course, what has happened is that Omicron has proven to be very, very good at eroding the anti-infection protection that comes with vaccination. So it can get into vaccinated people and get out. And it's been a real surprise to me how good it is at doing that. At the same time, the anti-disease protection has held up. That's a very important distinction. The protection you get against severe disease is being decoupled from the protection that you get against infection and onward transmission. So the variants have been very good, Omicron especially, at getting through the protection as far as infection and transmission goes, but the vaccines are still holding up against severe disease. Now, if this continues, that we still get new variants that are going to get around the vaccines and infection-induced immunity, then we're going to need to start thinking about some changes. Obviously, we're trying boosts at the moment. The vaccine schedules, the dose intervals, and indeed the doses are all a bit of a guess. We could try tweaking the existing vaccines, for example, combining them, so different mRNA sequences in the same vaccine. But I actually think what's going to be needed is next generation vaccines that are better able to target infection and transmission. And these might be, for example, vaccines that act in the upper respiratory tract and the mucosal immunity. So we might breathe those vaccines in, generate very good protection at the place where infection and transmission actually takes place. And there's also a possibility, as we see with some of the agricultural coronaviruses, that we have different vaccines all over the world, as we do with SARS-CoV-2, different types of vaccines being used in different places. And so in the agricultural world with coronaviruses, you can end up with different variants in different parts of the world. And so you need to modify your vaccines at different times, try different vaccines and so forth. We might end up with a situation with lots of different variants circulating in the world, and we need to use the vaccines that are most appropriate for the current time and place, as we do with flu vaccines, seasonal flu vaccines. So I think the answer to the question is yes, we are very likely to see other variants that come along that also, like Omicron, can get through into and out of vaccine-induced. People have seen vaccines or people have seen previous infections. And I really hope that the anti-disease immunity that we're currently seeing will hold up going forward. And historically, that has been the case. As I said earlier on, we're in new territory with this virus. We've never had anything like this before. But with a bit of luck, the anti-disease immunity will hold up and new variants won't sicken people substantially who've already been vaccinated or who've already seen the infection. So now I want to turn to the question of whether next generation variants, the ones coming next, are going to cause more or less severe disease. Now, by disease severity here, I mean, symptoms, and so hospitalizations and death, those sorts of severe symptoms. Now, it's really important that what I'm talking about here is whether the virus itself will become nicer or nastier. Because that is to say that the variants themselves are able to make people more or less sick. And so we can speak of a nicer variant or a nastier variant. So the common cold, for example, doesn't make us as sick as the flu. So that's a comparison of nastiness. Now, it's quite challenging to compare the nastiness of a variant. And in particular, this is the same plot I showed you before. When Delta was spreading in a population, it was making lots of people sick, but there weren't a lot of people at that point in South Africa who had been vaccinated or who had seen previous infections. And then when we've got a lot of people who've got vaccinated and seen previous infections, so got natural immunity, the variant then, Omicron, is now spreading in a population with a lot of immune protection. And so it's very difficult to compare and say Omicron is nicer than Delta because the conditions under which we tested them have changed. And ideally, what we'd like to do is compare two people who are otherwise identical, one with Delta and one with Omicron. Does Omicron make you sicker or less sick than Delta? And we can do those in experiments, as I'm about to show, but not with the human diseases, but in animal diseases. Otherwise, we are stuck really with trying to compare like for like, and that is really tricky. To me, the most important question is whether a future variant will cause more severe disease in an unvaccinated person who's never had COVID before. That's kind of the standard under which we want to compare things, I think. Although going forward, there's going to be fewer and fewer people in those categories. So we might end up saying, okay, what about unvaccinated people who've had 2 doses of a particular vaccine, but neither of them have had COVID before? In those situations, if somebody gets Omicron or Delta, which one is more sick? But I do think this issue of nastiness of variants is very, very tricky to compare here. Nonetheless, we'll give it a go. So this now is the same plot I showed you before, but I got disease severity on the y-axis and fitness on the x-axis. Remember, the natural selection is to push things to the right. So SARS-CoV-2 drops into the human population, and still a bit hard to know for sure, but roughly 1% case fatality rate when it first appears in the human population, right? When nobody has any vaccine-induced immunity and nobody's seen the virus before and recovered, so there's no natural immunity, drops in, it looks like it's in the order of 1% case fatality rate. Now, how does that compare to other coronaviruses? Well, SARS-1 had a case fatality rate of 15%, roughly, but it wasn't as transmissible as SARS-2. So it didn't spread as rapidly. And so it's got a lower transmission rate than SARS-2. So it's falling to the left, but up here. MERS, the Middle Eastern Respiratory Syndrome coronavirus, it has a very high case fatality rate, kills about 1/3 of people, but it basically doesn't transmit from people. It spills over to people from camels, but basically has no onward transmission. So it's got very, very low fitness in a human population. And then we've had coronaviruses for a while. So these are some of the viruses that cause the human cold. And so they are very little disease severity, mild colds and so forth. But we don't quite know how transmissible they are, but it seems reasonable to assume they're more transmissible than SARS-1 and not as infectious as SARS-2. So that's the kind of parameter space we have to work with the coronaviruses. And now we say, well, what's going to happen next in terms of the disease severity? And again, I want to emphasize that other things affect disease severity other than the virus type itself. Most obviously, people with comorbidities, older people, those things are more like a greater determinant of the chances that you'll die than the variant that you get. The virus genetics is a small component of the variation we've seen so far in who will die or who won't. But nonetheless, it's a question to ask, is in fact the subsequent variants going to be more or less nasty in a non-immune person? Controversial, but it looks to me like from the data we have are pretty compelling that Alpha and certainly Delta were causing more severe disease in completely non-immune folk than was the original virus. So those variants seem to have increased their virulence. Now, subject to my statement before about how hard it is to compare the virulence of a variant when the world is changing, we believe that Omicron is less virulent than Delta. So there's been some downward movement. And then the question is, can we find a variant in the future that is more transmissible on Omicron in the future world, and I've argued that we will get variants of concern. And so then the question is, will those variants of concern become more virulent, so cause more severe disease in non-immunes, or cause less? So that is what I want to spend the remainder of my time talking about, those three possibilities. And I'm going to focus on the upward one because the folklore is the downward one is most likely. We've seen all sorts of stuff about people have said Omicron is a sign of the way this virus is going to go, that it's going to evolve to become less virulent through time, because all viruses do that, because they don't want to destroy their hosts. They destroy their hosts, they destroy themselves. This has been such a predominant thing floating around actually for quite some time on Facebook. The PolitiFact have actually set out to try and debunk this. And there's a bunch of us being quoted in this article about trying to show that this is not the case. I'm going to come back to the theory, but I just want to say there's nothing in experience either that says viruses inevitably evolve to be nicer. Maybe before I just say that, one of the reasons for pause here is that SARS-CoV-2 doesn't kill very many people. And when it does kill them, it typically kills them long after transmission would have happened. So the majority of host death in SARS-CoV-2 is when the infections either would have cleared anyway if the host had mounted a good response, or the host die. So basically, from the point of view of the virus, at that point, the host isn't actually a home anymore. So right from the get-go, it seems like this might not be where we want to be thinking about in SARS-CoV-2. But let me give you some examples where viruses have not evolved to be nicer. This is a paper that actually came out just last week in Science, one of the world's most prestigious journals, showing that HIV is evolving, and we know that, but it's also showing that recently a variant that arose, so at the bottom there, a virulent variant that arose in the 1990s by de novo mutation has increased transmissibility, and it causes faster progression to AIDS. So HIV, which has been obviously in the human population now since around 1980, it is not evolving in any sense towards being a benign virus. The rabbit virus I mentioned is the classic case where people have said that the viruses evolved to be nicer. But this virus, when it was released into nature, killed 99.99% of the rabbits. Just think about that. Almost no rabbits that got it survived. That's an incredibly hot virus. In fact, no human viruses ever have been, well, rabies is like that, but it doesn't spill over. It's a spillover. It doesn't transmit on from us. No transmissible human viruses have ever been that nasty. And so, if you think to yourself, if this thing's going to evolve, it couldn't evolve to be even nastier. It's about as nasty as it could be. It can only evolve in one direction, and that's down. And Frank Fenner, who is now long past, but one of Australia's finest microbiologists, realized that when the virus spilled over and was killing those tens and maybe 100 million rabbits, that there would be very, very strong selection on the virus to become nicer, because it just couldn't persist if it kept killing every single host. And very rapidly, he pivoted his entire career to take viruses from rabbits and look at the evolution. And what he did was to stick the virus that he caught in the field into laboratory rabbits and see how they get on. And that is the equivalent of having the non-immune human to measure how SARS-CoV-2 evolution is proceeding. And so in the 1950s, he did experiments where he would stick the virus from the field into lab rabbits and measure through time the rate of death. His students picked that up again in the 1970s. And then for reasons that completely escape me, Australians abandoned doing that kind of work. And my colleagues and I picked it up in around 2015 or so, but using viruses from the field that had been taken in the 1990s. And this is one of Fenner's first papers on this. Within the past year, variants of the virus with diminished virulence have been seen. And he was incredibly prescient. And so the strain that he started isolating from the field was less virulent than the one that had been released. And this is a summary of what happened in that. This is an ancestral virus that got away, killed basically every single rabbit. And then Fenner's work showed that through time, the case fatality rates dropped. But I want to emphasize that this is still 7 out of 10 rabbits are dying from this virus. So what we call the evolution of more benign strains, this is still a lot hotter than anything we've seen in human viruses. But then as that work proceeded, the virus sort of seemed to hang around at this same sort of level through the 1970s, but then it started to escalate again. And the 1990 data are actually just one single data point that my colleague Peter Pugh collected, but it was a hint in the mid-1980s that things were on the move. And that's why we started this, got the finance to do these experiments, find out what was going on here. Now, I'm not going to go through all the data, but this is a death rate for a time for a group of rabbits. They're all alive at the start. You infect them with this 1950s progenitor, and 2 weeks later, they're all dead. This is one isolated in 1993. They're all alive at the beginning, and then 12 days later, they're all dead. So the virus from 1950 to 1993 got more virulent. It was killing at a faster rate. So it killed everything just like the ancestor, but it killed them more quickly. And what we discovered was that the modern day viruses, the 1990s viruses, have evolved the ability to immunosuppress the rabbits. They turn off the rabbit immune system, and the rabbits can't control their own bacteria. So bacteria which would live healthily in their gut or on their skin, kill them. And they kill them before the viruses can do anything. It's so lethal, this thing, because it's switched off the host immune response. And so it's absolutely more virulent than it was, and an absolutely ghastly virus. And I have to say, when we first saw this in the laboratory, we thought something else had got loose in the lab. And we threw the first corpses in the freezer, terrified that we had just unleashed something. This is just a natural virus that evolved in the field, absolutely destroys the laboratory rabbits in a way that they never have been before. And so the picture now is that this virulence is actually higher than it was before because of this immunosuppression. Now, why has that happened? Well, Fenner also started taking the rabbits from the field and looking at what the virus is doing in the rabbits. And we know from that work that the rabbits have evolved greater resistance through time. Okay, so if 99.9% of the rabbits are dying, any survivor is going to have a great advantage. So what's happened is that the rabbit got more resistant. The virus needed to overcome the resistance. And so it's evolved this capacity to immunosuppress the rabbit. And so if a modern virus meets a modern rabbit, you get regular myxomatosis. If a modern virus meets an old rabbit, instant death from this complete immunosuppression. In other words, there's been an arms race. It's just like a human arms race. The virus got more virulent, the rabbit got more resistant, the virus responded by getting more virulent, the rabbit got more resistant, the virus got more virulent. And this arms race is still continuing, it's still playing out now. And that accelerated evolution that we see in the 2010s in the Australian virus, we believe it's an escalating arms race that we're seeing. But most surprising to me was when I started to look at other situations where the same sorts of experiments have been done, and there's only a handful of them, and they're not as good as myxomatosis. But in every case where we've seen a spillover of a virus into a wildlife population, and then that population has evolved resistance, we've seen the evolution of enhanced virulence. So the variants that have popped up in all of these different settings have all been nastier than the ancestor. And we believe that's been driven by the fact that the hosts have responded to the viruses by becoming more resistant. They became more immune. So when I say there's nothing in theory or evidence, we have not actually documented, to my knowledge, a single case where there's been a spillover and it's got nicer. Now, I realize that's a bit weird because there are a lot of nice or moderately, pretty benign viruses around. So it suggests that it can happen, but we have never seen it happen in any documented case that I'm aware of, where we've been able to compare the virulence and look and say, oh yeah, here's a variant that's a more recent variant and it causes less severe disease than the ancestor. The one that I've actually spent most of my last 10 years or so on, maybe 15 actually, is a disease of poultry called Marek's disease virus. It's a herpes virus, so it's a big DNA virus, and it's shed from the chickens in the feather dander and it's inhaled. It causes cancer. And in the 1960s, it became such a problem that this sort of chicken farming, intensive chicken farming, wasn't possible. And if you're a disease ecologist, looking at all those birds in one place like that, where they're all the same genotype, the same immunological status, and they're all packed together, That is a beautiful place for virus evolution to take place. And of course it did. And so the virus got nastier in that setting and caused losses. And so they had to build, the poultry industry had to build a vaccine. The first generation vaccine lasted for 10 years, and then it was undermined by variants that could cause disease in the vaccinated birds. So there was a second generation vaccine introduced called the bivalent vaccine. That lasted 10 years. And then there was a third generation vaccine, Rispin's vaccine, and that has not been undermined by variants. And that is the reason your chicken nuggets are as cheap as they are. That vaccine is held. That evolution has played out in various different places in the world. And it is a rare example where virus evolution has led to the complete failure of vaccines that had to be swapped out and better vaccines developed. And I've been fascinated by that for as long as I heard about it. And so we have been doing experiments with different variants in vaccinated or unvaccinated animals, initially in these isolators, where we put birds in there, they grow up, spend the life there, they go from being these little chicks to being big birds. And the transmission happens naturally in these isolators. Did this work in the UK with colleagues from the Pugh Institute, particularly Virulence, and the containment facilities are very tough for these sorts of experiments, so we can only do them in a few places in the world. Later in our work, the European Union started regulating, we do it in floor pens, so our work then transitioned to these floor pens, which are perhaps a more natural transmission setting. We worked with 5 variants. This is one of them, this B14. This is a survival curve through time. And so you start off with 20 birds, and after 2 months, half of them are dead if there's no vaccination. If you vaccinate those birds, all of them are kept alive. Okay, so this is still a pretty nasty virus. It kills half the birds and it kills them in 2 months. But the other viruses we work with, this one kills most of the birds in 10 days, and a few hang on and then die after 4 or 5 weeks. Here, most of them killed, but then a few hang on for 3 weeks. Here, they're all dead in 10 days. And now we're starting to get deaths in the vaccinated group. So these variants, these virulent variants, kill unvaccinated birds, and they also start to kill vaccinated birds. So these variants are these more severe disease-causing variants, much more severe than the ancestors. And these were isolated from farms. And in the 1960s, when B14 was isolated, nobody would have conceived as 675A. Nobody could have imagined a virus that was capable of evolving from B14 and killing all the birds in 10 days. It's quite clear from the literature. Nobody could see that. Nobody talked about it. Nobody imagined it. But over those 30 years, variants popped up which were able to be that lethal. And I've been fascinated by how that is the case. We've been doing tons of work with those 5 variants in vaccinated and unvaccinated. I just want to show you one experiment with one of the most lethal strains. We mixed birds that are experimentally infected with that lethal strain with naive sentinels. So we put 20 birds in there. Half of them are infected with this variant, and half of them are just naive. Nothing's happened to them, and they just live with the other birds. And when we do that, all of those experimentally infected birds are dead by day 10. Dead, I have to say here, is euthanized. So we can tell pretty well in advance when they're all going to die, and so we kill them before things get too gruesome. But the death proceeds pretty quickly, and maybe they would have survived another day if we hadn't put them down. But all the others were alive, and we know that they didn't get the virus. We know because we can measure the virus in the dust that's in the containers, and we know that these birds never got exposed because the virus wasn't shed from these birds before they died. Whereas when we vaccinate with the first generation vaccines, we keep the birds alive, and in that case, they stay alive because they're vaccinated, but they transmit the virus to the sentinels, and all of them die. So these birds were kept alive by the vaccination. They were able to pass the virus over to the other birds, which then died. And if you look what's going on in the birds, so this is the virus titers through time in vaccinated birds and unvaccinated birds with this benign variant, then the unvaccinated birds survive because it's not too nasty. The vaccine suppresses the virus by 100-fold or more at any point in time. And so in this situation, then the unvaccinated birds, the virus is coming out more from those, but it's suppressed, but it's still able to transmit from the vaccinated birds. The very virulent ones, the unvaccinated birds, die in 10 days before sufficient virus has been shed. If you then vaccinate them, keep them alive, they are able to transmit because they are kept alive and the virus is shed from them into the environment. So vaccination keeps the birds alive and makes it possible for these hot strains to spread to other birds. So strictly speaking, we do not know why these hot strains appeared, what caused them to evolve, but we do know that they can, however they evolve, whatever caused them to evolve, they can only be transmitted and maintained in vaccinated populations because they're too nasty to spread otherwise. Now, such a thing is not possible in SARS-CoV-2 at the moment because we don't have anything like that level of mortality. But I think the way this sort of evolution could proceed in SARS-CoV-2 is that we know that when people get sick, they stay at home, isolate, don't go to work, don't spread things around. And that we encourage people to stay at home so they don't infect other people. We know it interrupts the transmission. That's the whole point of isolation and quarantine. Now, if you vaccinate people and they are still spreading the virus, but they're no longer feeling sick, they won't stay at home, they'll spread the virus. And we'll have a situation where strains that would previously not have spread because of the symptoms are now able to spread. And I think that's a distinct possibility for the future in SARS. So summarizing all of that, I think it could go in the future anyway, and it might go all ways. So I've talked about myxomatosis, where things went down in the first instance, up later, but down first. We might see something like that. Diphtheria and pertussis are examples where things went down. Flu, every year flu seems to be, at least from the same type of flu, seems to be roughly the same level of disease severity. Typically, it's a new pandemic before we get changes in severity. So it seems like flu virulence just stays much the same. Or it could go up, like I've just shown you in Marek's, although I realize pertussis is in both these cases, whooping cough in both cases, there are variants in whooping cough which make the bacteria less virulent that have spread in vaccinated worlds. And there are different variants that have spread where they cause more severe disease, although still very mild. And everybody is still better off in those cases being vaccinated. And from the point of view of how the variants in SARS-CoV-2 are going to proceed, it really matters how disease severity affects transmission. If disease severity restricts people to the home, cuts down on transmission, then more benign variants would have an advantage because people will go out in the world and not feel sick and they'll spread it. If, on the other hand, people are protected by vaccination from feeling the effects of severe disease, they'll go to work despite the fact they've got a strain which otherwise would have kept them at home, and they'll spread that. So it's really an open question, and it could go, I think, either way. I know that's not a very satisfactory explanation, but I do not think that there's any way we can say in theory or in experimentation until we know what's going on, which way it will go. We can't predict it. So I think we should hope for the best. I think we should hope we get variants that are highly transmissible but cause almost no disease because they'll immunize the world. But we could also get a situation where we get something like what happens in Marek's. And I just want to say in the Marek's case, what happened was, and the poultry industry is doing very well, despite the circulation of these viruses, what's happened is that new vaccines have been developed which have allowed the industry to proceed despite the vaccination. And I think that's what will happen in SARS-CoV-2, that we will need second and third generation vaccines that do a much better job at stopping transmission should more disease-causing variants come along. And remember, in vaccinated populations, such variants wouldn't necessarily make vaccinated people or immunized people very sick. It's people who would be not able to get vaccinated or have immune responses that are suboptimal. Those people would be at greater risk. And Nicole, if I can just have 2 seconds more, I just want to say that our work on the chicken vaccines has been taken out of a lot of context. This is actually our paper being shown on Joe Rogan. I'm not sure if Joe Rogan is the same following in Canada that he does have in America, but Joe Rogan is one of the podcast shock jocks. And he discussed our paper in, I think it was October of last year. And these are the views of our paper from when we published it in 2015. This is the Rogan effect here. 500,000 people have now looked at our paper. 500,000 people. I find it incredible. And actually 20,000 people, 25,000 people have downloaded the paper to read it. This is a scientific paper. I mean, it's incredible. And it is now running at number 69 of all time on social media out of 20 million papers being followed by Altmetrics. Actually, we're also number 70. So if you add the two together, it's very, very high media, social media coverage. And that has been because people have been saying you shouldn't vaccinate because you never know what's going to happen with the future evolution. And what I really want to emphasize is here that I just said we don't know about the future in this case. We can't predict what's going to happen next. So I've just sketched out scenarios, and any of those scenarios could happen. Why it is that people want to hold back life-saving drugs because of evolutionary hypotheticals I've never understood that at all, especially given that should something like the Marek's disease case play out, we can deal with it. I also want to say that we don't know whether vaccine-induced immunity and infection-induced immunity actually do much different here or the same thing. And that's a really critical thing. People who don't get vaccinated will get infected. So they too have immunity that Omicron can get through. So that immunity is shaping the evolution of the future too. So right now, we don't know, and I don't think we ever will know enough to be able to modify our current vaccine practices out of fear of future scenarios. We should try to block transmission, but the idea that we might somehow take evolutionary predictions which we really struggle to make and change how we do medicine now just seems very unlikely to me. So emphasizing that the crystal ball is cloudy and we don't know what's going to happen next, I think we should hope for the best and think about and plan for the worst. And with that, I'll say thanks very much, Nicole.
Thanks, Andrew. You'll have to accept my applause from 360 some people. That was a fantastic talk. Hello, everyone. I'm Nicole. I'm an associate professor in the Department of Ecology and Evolutionary Biology, and I will be your question moderator this evening. So please feel free to throw your questions in the Q&A. I'm going to start things off. I'm going to take my moderator prerogative here and start off with a couple of questions. But I'm going to go way back to the beginning of your talk, if you don't mind this detour. So your very first slide, you showed us the phylogeny. And I wonder if you can do two things. One, just very briefly for folks who aren't used to looking at phylogeny, say something about what that is, what the data is that produced that. And then my second, part two of that question is, it feels like there's a lot of data. So I take your point that looking at a whole bunch of birds doesn't help you predict the kakapo, but we've got a lot of birds here, right? That looks like a ton of data. So I wonder if you could just comment on sort of the situation we're in with this pandemic and the sort of abundance of data.
Yeah, well, so the first thing, that phylogeny is a tree of life for the virus. And so that's the relatedness of, as you think of it, just like your own family genealogy. You had a great-grandfather who, married people and made more people. You get a tree. That's the same. That's the tree of the virus. And we get that from the sequences, the genetic sequence of the virus. And so mutations build up in the virus, and you can see from the mutation patterns who's related to whom. So that's where that comes from. We do have a bucket load of data. There's no question we have more data on this virus than any other organism, I think, ever. And it's a really exciting time if you're an evolutionary biologist, because this stuff is so freely available. It's incredible. Yeah, I still put it to you that no amount of sequence data will allow you to predict what's going to happen next until it's happened. And that's because of this very challenging question of trying to predict what's possible when all we can see is what's already happened. I also think, as you know, Nicole, that one of the biggest problems in biology has been mapping genotype to phenotype. And just to put that in perspective, people have known how to sequence flu viruses since the 1970s. And seasonal flu vaccines are all about predicting what variant is going to be around next year. And so we've had these sequences. We've known we go take a look in the southern hemisphere, and that's how we try to predict what's going to happen next, what's coming for us. We can't tell from the sequences of what we've got now what's going to be a big deal next year. And we've been trying to do that with these great flu data for a long time. So it's not a question of the amount of data, I think. It's a fundamental question of, reason we know about the birds is we've had whatever it is, maybe hundreds of millions of years, tens of millions of years, just trying to get that right, tens of millions of years of bird evolution, and we think it's kind of exhausted. We think that there's nowhere we've missed anything. But if you were back there at the ancestral state of birds in year 2, trying to predict what birds would be like, and I put it to you, even with all the knowledge what the Europeans had, until they saw the kiwi, nobody conceived of it.
Cool. So my second question is kind of follows from that. And you mentioned you put up that Nature paper with your buddy Eddie Holmes, and you pointed us to the quote about mutations rarely impact outbreaks dramatically, and we talked about how that didn't age very well. And definitely, it's the case that some mutations have impacted outbreaks dramatically. Yes. But is it not the case? Is it true that they didn't say it? They never impact outbreaks dramatically. So has it not been rare? Like that phylogeny shows a whole bunch of mutations that didn't lead to surprising new spread.
Yeah, that's right. The vast majority of mutations damage the virus, and so they don't go anywhere. And it's only a tiny minority of the mutations that give a benefit, and often it's a tiny minority of the combinations. Nonetheless, I put it to you that the dynamics of this COVID-19 have been completely changed because of the evolution of these variants. So Omicron and Delta and Alpha, these are super rare events, but they completely transformed the outlook. And going forward, I think that's going to continue to be the way. The strange thing about that paper, rereading it yesterday, they actually talk in there about things that have really mattered. For example, West Nile virus, which is one of the ones in my example, that spilled over into North American birds. And one mutation drove it through vast numbers of birds and killed horses and all sorts of things. And so, I think at the time there was a lot of discussion about coronaviruses not mutating very much because they have quite good repair mechanisms compared to other RNA viruses. That's in a world where mutations are limiting. When you've got tons of viruses, every mutation is expressed, right? And so, yeah, I was a bit startled at the time that we were going to be so confident about that. And, flu's dominated by the evolution as well. So I think it was a lot of optimism at the time, which has not stood the test of the next subsequent 2 years.
Yeah, fair enough. Rare things become not rare when you have multiple opportunities, many opportunities. So I'm going to turn it over to the audience. And while we're on this topic, I think one of the questions is kind of related to this. Could you please explain the difference in terms of severity and fitness and evolution between annual flus and COVID?
Yeah, at some deep level, I'm not sure that anybody can actually. We have, so the superficial answer to that is it depends on which cells things get into, what they do in those cells, and how the immune response deals with them. And sometimes the immune response is, alarmed by what's going on, and so it overreacts, and that overreaction can cause severe disease. And oftentimes that's what's true in severe flu, that people have what are called cytokine storms, which is an inappropriate immune response being mounted. We are still unclear about what's going on, I think, at any deep level, why some people seem to clear COVID without too much problems and other people can get severely ill and die. And that's presumably tied up with where the virus has got to and how the body has responded to it. But at some deep level, I'm not really sure that we really have compelling data on that. Obviously, one of the things people talk about with Omicron is that it's an upper respiratory tract virus. And to the extent that we think Omicron is less virulent than previous variants, that's a possible explanation. It doesn't get it deep into the lungs, and so it doesn't set off immune responses or do damage to lung tissue, so it's easier to breathe and so forth. I still feel like I think we'll have a much better understanding of this in a few more years. Right now, I think there's quite a lot of hand-waving going on.
Sure. In terms of mutation rates, we know some stuff about COVID versus flu, SARS-CoV-2 versus flu, right?
Yeah, that's right. Although, of course, the mutation rate per virus, we know that there's so much SARS-CoV-2 out there at the moment compared to flu that even if the mutation rate was 10 times slower in SARS-CoV-2, there's way more than 10 times as much flu. So the net effect is evolution on steroids.
Absolutely. So you mentioned, where the virus is infecting can change the severity of infection. Thais wonders, are there other known strong predictors of increased health impacts on the host for the virus? Maybe something about the virus family or how it's transmitted?
So I think that's a really interesting question about what can we learn from other things, right? And initially people would said, well, it's okay, it's going to be fine because the current human coronavirus has just caused mild disease. It's kind of like, okay, but SARS-CoV-2 doesn't. It causes severe disease and not as severe as SARS-1. So then you say, okay, what about other coronaviruses and other families? Well, there are there are dog and cat and chicken coronaviruses, pig coronaviruses. Mostly they're pretty nice, but in some of the chicken ones are pretty nasty. And so until SARS-1 came along, there weren't actually people studying, many people studying coronaviruses in humans because they were uninteresting. So I don't think we can learn much from other human coronaviruses. So I'm not sure we can extrapolate very easily based on relatedness. And I think that's been an eye-opener. And so to me, as people have said, well, the chicken virus is a very different virus from SARS-CoV-2. And I agree, it's genetically very different. Is that the main issue? Or is it the fact that it gets through transmission, it can transmit through vaccinated hosts? Is that the main one? Pick your analogy. It's very early days here, I think.
Okay. If we have a question from someone who wants to know, when we're talking about sort of disease preventing vaccines, you mentioned the kinds of severe symptoms you were talking about. Hospitalizations and deaths is what we were often talking about. But this person would like to know, to what extent does that apply to long COVID, and to what extent should we be considering that a severe outcome as well?
Yeah, I think it's the case. We still don't have sufficient data, I believe. But I do think that the data we do have suggests that by and large, the virus is over and done with in a few weeks in terms of transmission, that the transmission windows are happening, most of the time it's 2 to 3 weeks. Sometimes the window is 4 weeks, maybe 5 weeks. And long COVID is associated with damage. But it is possible that there are grumbling chronic infections going on there causing the long COVID. So there are grumbling viral infections. We see that in other viruses. We have, chronic grumbling infections can keep people sick for a long time. I think whether those cause transmission is open to question at the moment. And if they don't cause transmission from an evolutionary point of view, that doesn't matter at all. Obviously, it does matter to the person who's got long COVID. But at the moment, it's very hard to imagine that a substantial amount of the transmission in the world is coming from long COVID patients with long COVID. I think that's going to be a very tiny, if any, proportion of the amount of transmission. It's all coming from the acute infections. Mm-hmm. So at the moment, anyway, the evolution is being driven by the acute infections.
So we have a question from Madeline that's about the Merrick's vaccines. And she noted that the deployment of new vaccines seems to come right at the tail of failure of previous vaccines. Are these vaccines very similar in composition? Is the red tape in vaccine development for chickens just lower, much lower than for humans? What accounts for this rapid turnaround?
Yeah, so it is easier to bring out a chicken vaccine than a human vaccine. Although the speed with which we got these RNA vaccines is pretty close to the speed with which the poultry industry can generate a new vaccine. In poultry industry, they're allowed to make vaccines themselves and pretty much at a company level. And so depending on how it's being marketed, there's almost no checks on it initially. So they can respond pretty quickly. Of course, it's also much easier to do the experiments with animal vaccines because, you've got the animals, they are contained, you can compare like for like. And in the case of chickens, you can do thousands and thousands of animals in a hurry. So it's much easier in principle to develop them. The reason it took 10 years each time was not because of the difficulty of developing the vaccines. It's just the first one worked so well, nobody felt the need to do anything more. And I have heard some of this sort of talk at the moment in the case of the SARS-CoV-2 too, that we've got these fantastic first generation vaccines. They stop severe disease. Why would we be interested in second or third generation vaccines? And to me, it's to stop the transmission. So even if the anti-disease properties continue, we need transmission blocking vaccines, not least for the people who can't get vaccinated or can't respond to them. And so in my head, that's second, third generation vaccines are going to be things like these mucosal vaccines that are we hope, much more effective against transmission. Cool. And there are 200 or something vaccines still in development, right? There's a lot of work going on the second and third generation vaccines.
Yeah, it's quite an impressive amount of work and
it is.
On short timescales, as you pointed out.
Okay.
I've got two questions that I'm going to try to merge together. One from Amar. I realize that it's hard to predict exactly what sort of variants will emerge, but could you hazard a guess on the rate of emergence of new variants? And related to that, we've got a question from I know this is true, an evolutionary biologist, PhD student here. Given the huge population sizes of the virus and the relatively high viral mutation rates, what do you think limits the waiting time between successful variants?
Yeah, so hang on a minute. I've just put my neck out and predicted where things might go and what new variants. And now you want to
know
when.
Yeah.
Okay. For the reasons I said at the beginning, there's a lot of contingency involved here, folks. So we know now that every point mutation, that's to say every base pair change possible in the genome, has been expressed somewhere in the world. We know that many combinations that are possible have been expressed somewhere in the world, but the number of combinations across the full genome is immense. And even if a variant pops up, what happens next is critical. And so if it doesn't get out of that person, or that person doesn't then infect, go to a party or whatever, it can die out. And so most of the time, the new variants are popping up and dying out, popping up and dying out. So that stochastic effect is like tickets in the lottery. And I think that actually makes it very hard to predict. What we might say is that there are conditions which are going to favor the spread of the virus, and those might be associated with more variants, like a variant that pops up in the run-up to winter, where people are coming indoors and in high, that's going to have a big leg up compared to a variant that pops up in early summer where people are outside. I think that sort of seasonal forcing might become a deal, as it is with flu. But I don't think I'd want to say much more about that. I mean, Omicron, it apparently took the best part of a year before it got going. So it was hanging around somewhere, and then it's off. Right now, was it hanging around in an immunosuppressed human or an animal population or one person?
Yeah, so we had a question from an attendee about exactly that. What is of those options? What is your speculation about? What's the best guess for what?
Yeah, I'm very nervous about the animal population side of things. Presumably, you've heard in the media the coverage about the deer populations in the US, and I guess that must be true in Canada too. Just don't know how much work's gone on up there. But in Iowa, there were more deer with COVID at any point in time than there were people. And it's everywhere. It's a hugely high proportion of the deer, including the ones just walking across my backyard, have COVID. And we've already seen from the sequence work some of my colleagues have been doing here at Penn State that those viruses are evolving into deer. So there are deer-specific lineages evolving. Now, they probably get pretty good at deer, and let's hope they get less good at humans while they do that. But that creates another thing that can recombine with the ones we already have. So that gets me worried. Probably my biggest concern actually is that the situation which gave rise to this thing in the first place was a spillover. That's happened 3 times a century, right? SARS-1, MERS, and SARS-2. So if that's happening every 7 years, a spillover from wildlife is getting into humans and getting going. Now, if it's spilled over at the moment, while we have bucket loads of humans infected with SARS-CoV-2, got a recombination between something new and SARS-CoV-2. I'd love to get those vaccines rolled out into all areas where people are coming in contact with wildlife. I think it's absolute amazing tragedy that it's like ignoring potential war zones. There's nothing that's changed that makes this event, the spiller event, less likely to happen now than it did when it first happened in 2019. Yeah.
What are people thinking about vaccinating wildlife?
So that happens in other situations, like rabies vaccinations are possible. I think the interesting thing in the deer population is I'm told around here the average deer lives 1.5 years. And so there's a huge amount of susceptibles coming into the population all times. And so it's going to be quite different from the dynamics in the human population. It's hard to see how it would ever get eradicated from a deer population. Even if we could vaccinate the deer, they'd all die soon, and then we'd have a bunch of unvaccinated deer. So that's just one animal species, right? So,
yep.
Mind you, it'd be probably easier to vaccinate the deer population than, because we can control it, right, than the human population, which we can't control.
Yeah. Deer are all for vaccine mandates.
Yeah, and masks.
We have a question from Nick Martin. Will next generation vaccines, which focus on parts of the virus which have less mutability, be more effective in long-term protection?
Yeah, so that's a question that's relevant to all types of vaccines. So I've been working a lot on malaria vaccines, and people have, which have by and large not worked very well because of this problem. People have always argued if you've confined yourself a part of the virus that doesn't have lots of variation, then that's the bit you need to glue the vaccine onto, so the antibodies onto, and then you'll have better. Of course, the reason it doesn't have lots of variation is because immunity is not naturally seeing it. If it was naturally seeing it, it would either be wiped out or it would be polymorphic, variable. So you could say, well, we could target a conserved region, a region that's not changed much, and that might work great. Or as soon as you target it, it will no longer be conserved. You have to find the right balance of something that just simply can't be mutated for structural reasons. And I think a better bet actually would be to target multiple parts of the genome. And at the moment, it's crazy to me that we're still focusing just on spike. I mean, it's amazing we've got away with it as long as we have. I'd like to see, various different spike sequences in the mRNA vaccines, but also some non-spike proteins in there as well. So.
Yeah. Does flu provide another good analogy here where surely we've been looking for universal flu vaccines for a long time.
But Nicole, there are people that think they're on the cusp of a universal flu vaccine, right?
Maybe we are.
And maybe we are. And I mean, we should look at those conserved regions and see if they are really immutable, can't be changed. Yeah.
So you probably know, maybe we've kind of really stopped testing in Ontario. There's very limited PCR testing. And so someone would like to know, does a lack of testing in a population make your crystal ball even more cloudy?
I think I've begun less and less interested in the human testing data, partly because it's really a reflection of human test-seeking behavior and human test availability. So it's a better measure of what humans are doing than it is of what the virus is doing. And I don't know about Ontario, but certainly here, people have mostly moved to at-home testing and antigen testing. That's not reported through any national system. And so we actually have no idea what's going on, even in State College here. And we're looking at in the wastewater, and that, I think, is a better reflection of what's going on than any test data. I would say that the lack of testing does have one effect, because it means that there are people who, they lost a second way of knowing they were infected, right? One way was that you were sick, so you'd stay at home. Another one is that you tested positive, so you'd stay at home. Now we've got vaccinations to stop the first way and a lack of testing to stop the second way. So now there are variants able to be moved around in the population because nobody knows they have them. And the silent spread of variants, I think, is a very real prospect in an immunized world where the immunity is not stopping transmission.
Yeah. So someone has asked about, sorry, I've lost it in this sea of really fantastic questions. So I know I keep talking about flu because I think flu is super interesting, but Pete would like to know, H1N1 was very lethal to begin with. It was a pandemic flu virus, but now it's just one of the possible annual flus. Can you talk about that process? Is that an example of a virus becoming nicer?
Yeah, so just let me go back. We made that comment earlier about when you can extrapolate from one virus to another. Mm-hmm. And it's interesting to me that all the people who've studied flu say one thing, and all the people who've studied other viruses say different things. And it really is interesting whether or not flu is a good analogy for SARS-CoV-2 or it's a misleading analogy that's getting in our way of thinking. And I think the jury's out on that still. Don't have strong view. You don't see in flu variants that simply have a transmission advantage, just, take over. They have an antigenic advantage. They're different. So it wasn't flu becoming fitter and fitter through time, like we've seen at least up to Delta. I don't think we actually know with the H's and the N's, the different, when we get a new combination, so a new pandemic of flu, a completely new combination of H's and N's, or one that hasn't been seen by humanity for a long time, it's moving through a non-immune population. It quite quickly generates an immune or semi-immune population. And so the impression is that the virulence goes down. And I really don't think we know for sure whether or not that's because of virus evolution or because of the immune populations become more immune. If you look at other things like avian influenza, which is, a flu, what happens there, we have a thing called high-path AI. And high-path AI is a mutated form of low-path AI, and high-path AI mutates, and we know the mutations in many cases, and those are lethal, those strains, due to the mutation that's known. Like the variants are thrown up, and they're typically so nasty that not only do they kill all the flocks they get into, but people kill all the flocks in the area to wipe the thing out. We had one of those in turkeys in the US last year. And so that's a case which I think is, you can do the experiments because you can put those two viruses into the chickens or the turkeys and compare them. With the seasonal flus, we really struggle a bit, I think. Yeah, that's a bit of an unsatisfactory answer, and I want to say it's because the flu guys don't really have the right data to say.
Okay. I want to like push you to maybe say something about policy, perhaps. So given everything we've learned so far, Robert would like to know, would you have any advice for governments as to a better reaction to some novel infectious agent that might arise in the future?
So my general comment on this one is that we had 2 warnings this was going to happen, and a bucket load of people saying it was going to happen, and governments and everything planning. And we, as it's not just America, although man, oh man, performed very poorly under a known threat. And I think the general issue is we need the equivalent of an army. It's a long time since America got attacked, but because of Pearl Harbor, systems are in place to keep a good eye out. And most of the time, the people involved in that, the hundreds of thousands of people, sit around doing very little except practice. And we need that sort of investment and that sort of talent practicing all the time for these sorts of things. My biggest concern, and I see it even locally here, is that people think we're over the worst. We'll wind down the testing capacities. We'll not push the vaccines as much. We'll all get complacent, and the expertise goes away. And so my general comment on that is that we need to keep, that's what I meant about hope for the best. And I'm with you. It's all hope for the best, but let's plan for the worst. And we do that with wars, and we should do it with things that are incredibly damaging. More Americans have died, and I assume more Canadians, more Americans have certainly died from COVID than had died in all the wars. I mean, it's insane. We spend squillions of dollars on protecting against wars. I mean, it's just incredible to me. So even locally, I've seen that the testing capacity being wound back, the facilities, the IT systems that it turns out to need to know who's got vaccinated and how you get the systems going, all of the sort of systems that manage to get us to roll out vaccines in record times. We need to keep those capacities running, even though we're not using them, so that if a variant comes along, which really does get around, it does cause severe disease, and we need to get it all cranked up again, it's there, boom, not the 9 months, 10 months. It's amazing to me, we got these vaccines and then we couldn't use them properly.
Yeah. So another attendee wants to know, it points out the recent Lancet article that said, while COVID is here to stay, the pandemic is soon to end within months. Are your thoughts, let's hope for the best? I think that's a direct quote that you just said. We'll hope for the best, but prepare for the worst. Yeah. What are your thoughts on the pandemic ending within months?
Yeah, so I think actually pandemic and endemic and stuff are not helpful phrases here. This thing is with us forever. And other diseases that are with us forever that are we would call endemic, like malaria and TB, those are endemic diseases that kill millions of people every year. So the idea that what we mean by the pandemic over is maybe this sort of out of control, crazy ass doubling times of 2 days are maybe going to be something rarer. I hope so. And that's what I'd hope for the best. But even if they're grumbling along and the variants are coming and going, and so the doubling times are every week or 2 weeks, we still need to pay attention. I mean, I just think this, I know we're all exhausted and we'd like it to finish. I keep thinking what it must have been like in the middle of Second World War. You have no idea how long it's going to go on for. And in retrospect, we know it all stopped in 1945. And we just want our 1945 moment, but they didn't have that until 1945 when it stopped. And I'm not sure we'll ever have this stop. I think what's going to happen is that people who are not able to mount full immune responses will always be at risk for this thing. And the rest of us, with intact immune responses and so forth, be okay. But those people on chemotherapy or the elderly, they'll need to have special measures and care going forward, because this will still be a significant risk to a minority of people. Yeah. If we get a variant that causes more severe disease, that risk would go up. And if we get a variant that can escape immunity, and especially anti-disease immunity, it's going to be more serious again.
Yeah.
Yeah. I hope for the best, folks. Let's hope it melts away, just like the 1918 flu did. Let's hope it's different. It's the same as that. I mean, I really do hope so.
So Emmanuel wants to know, is it reasonable to assume that the variants would be nicer in parts of the world where vaccination rates are low?
So there's a very important distinction there between the variant, two variants that differ in their virulence, and how that difference plays out in a person that's vaccinated or unvaccinated. So it could be that there are more virulent variants circulating around, let's say in North America, but they play out less virulently in vaccinated or people that have seen immunity before, because they've got immune protection. Likewise, you could have a situation where a pretty mild variant is circulating in a place where there's not much immunity, and it's devastating. So I really get nervous about this, trying to figure out, there was actually questions being asked, seriously, the conference I was at, should we vaccinate some countries or just leave them unvaccinated? And I just find it appalling that we would be in a situation where some sort of evolutionary scenarios, which I've just told you is a crystal ball, it's cloudy, would allow people to make decisions about not vaccinating, not giving life-saving vaccines to people. I mean, I just find that totally bizarre, to say nothing of the fact that virus circulating in populations is a source of evolution of new variants. So we really should try to get on top of things. I really think we need to decouple evolutionary risk from short-term benefits. And like I say too, if this thing's coming from wildlife disrupted environments, let's get those damn vaccines in there and let's get those people protected. And maybe they need masks and maybe they need to be given other jobs so they're not torturing, pulling animals out of the forests and exposing us to this risk. I mean, maybe we need just to invest in all those people trading in bats, find them something else.
Okay, I'm going to ask you one more question. I know I'm leaving a ton of questions unanswered in the Q&A, and so I apologize to all you very enthusiastic people who are asking amazing questions, honestly. So my final question, do you have any short and simple takeaway messages that science students and others can use to explain to their non-science friends and family? What are the most important takeaways for folks from this talk? And
the first thing I want to say is I have enough trouble understanding virus evolution, and understanding human beliefs and behaviors is not something that life scientists do, right? That's a question for people in communication science and sociology and behavior change and so forth. Yeah, but I guess the simple message I would say, personally, the way I feel about this, that's maybe the best way of thinking about it. I personally feel a lot less at risk than I was because we have fantastic vaccines. I personally feel that I've got a good eye on the situation, and should things, unpleasant variants appear, I can go back to working remote and keep the distance and all of that sort of stuff. So I'm feeling pretty optimistic about how my summer is going to go. I really feel for the people who don't have good immune responses. And that's not just people, that's people who chemotherapy for cancer, older people, underlying comorbidities. There are people, we've been doing a lot of antibody testing, and there are people who get vaccinated and, friends of mine just don't mount a response. I know I have, and I've been checking it. But, I feel for those people. They're at a different level of risk. And until we get vaccines that stop transmission, or we get some sorts of, lucky break, those people are always going to be at risk. And so once if the virus is around a lot, start modifying and you're at risk, start modifying your behavior and pull back.
Okay. Thank you for answering all those questions. So to the audience, I just want to say thank you all for coming. It's been a really interesting night. I hope you've taken something away from today's talk and the questions. Just to let you know, we're going to be sending you a survey. You'll probably get it sometime before 9 p.m. tonight. We're sending it out right after the session. If you wouldn't mind responding to that, we'd greatly appreciate it. We have also recorded this session, and that will be available, and you'll get an email about how to access that video as well. And one last thing to say, thank you, Andrew, so much for taking the time tonight. It's been really fascinating. Thank you for walking us through the past and giving us an eye on the future.
My pleasure.